To understand how an entire species vanishes, we first have to map its environment. Christmas Island is an ancient limestone seamount sitting in the Indian Ocean, isolated by a massive 1,500 kilometers of open water from the Australian mainland. This extreme geographical isolation created a unique evolutionary laboratory. When British naturalists first surveyed the island's primary rainforests in 1888, they documented a thriving population of the Christmas Island shrew. The endemic mammals were so abundant that observers described the night forest as constantly echoing with their sharp, bat-like squeaks. Within a span of just 20 years, however, the forest went entirely silent. By 1908, the shrew had effectively vanished. Decades of sporadic searches followed until October 2025, when the IUCN officially closed the book on the species, moving it to the extinct category. Overhunting and direct human culling play no role in this timeline. The loss of Australia's only native shrew is a strict forensic case study in systems ecology, demonstrating exactly how isolated environments undergo compounding structural collapse. To identify the vulnerability of the species, we have to look at its thermodynamics. The Christmas Island shrew belonged to the family Crocidura. Because shrews are incredibly small, they possess a massive surface area to volume ratio, meaning they lose body heat rapidly. To compensate and survive, their metabolism operates continuously at maximum capacity. This comparative chart shows the daily caloric requirements of the shrew against a baseline mammal of similar size, like a standard mouse. To avoid starvation, a single Christmas Island shrew had to consume between 1 to 3 times its own body weight in insects every single day. Meeting this massive energy budget forced the shrew into a state of near-constant frantic foraging. They required a specific microhabitat to survive, the deep, complex layer of humid leaf litter on the primary forest floor. This thick layer provided a constant supply of beetles to eat, while simultaneously keeping the shrews hidden from larger predators. The shrew existed on a razor-thin metabolic margin. Any disruption to the leaf litter directly severed its food supply and its structural cover, guaranteeing immediate starvation or predation. The first disruptive variable arrived in 1899. Human shipping brought stowaways to the island, introducing invasive black rats to the pristine ecosystem. These mainland rats acted as living vectors, carrying a flea-borne parasite known as Trypanosoma lewisi. Christmas Island already hosted 2 native endemic rat species. However, because they had evolved in total geographic isolation, their immune systems were completely naive. They had 0 evolutionary defenses against mainland pathogens. The resulting spillover event was remarkably swift. Within 4 years of the parasite's introduction, both native rat populations had crashed to 0. The elimination of an island's apex native predator creates an immediate ecological vacuum. The regulatory pressure keeping the lower tiers of the food web in check disappeared overnight. The dominant native rat species had historically fed heavily on juvenile red crabs. With the rats wiped out by disease, predation pressure on the crabs vanished entirely. Left unchecked, the Christmas Island red crab population exploded into the millions, aggressively expanding their footprint across the island's terrain. Red crabs are terrestrial herbivores that consume fallen foliage. The artificially inflated population required massive amounts of calories, and they systematically stripped the forest floor entirely bare of fallen leaves. This cross-section models the physical impact on the environment. As the crab population line spikes upward, the deep leaf litter layer vanishes. The shrew's terrestrial niche was effectively deleted. Stripped of cover, the remaining shrews were starved of invertebrates and exposed to feral cats and snakes introduced by human settlers. The introduction of a microscopic blood pathogen in one mammalian species ultimately destroyed the physical ground cover required by another. It is a precise illustration of a compounding trophic cascade. The second phase of collapse arrived in the 20th century with the yellow crazy ant. Unlike native species, these invasive insects cooperate across different nests, allowing them to form unicolonial supercolonies spanning hundreds of hectares. This systems model illustrates the mutualistic feedback loop that triggered their population boom. The crazy ants farm another invasive pest, the yellow lax scale insect. The top half shows the scale insects sucking sap from the forest canopy and excreting a sugar-rich liquid called honeydew. The bottom half demonstrates the ants protecting the scale insects in exchange for this carbohydrate supply. Bypassing normal ecological limits, the feedback loop accelerated rapidly. Ant density swelled to an unimaginable 1,000 ants per square meter. Yellow crazy ants do not sting. Instead, they overwhelm prey by spraying a blanket of blinding, immobilizing formic acid from their abdomens. Any shrew that survived the trophic cascade now had to navigate a highly acidic wasteland. A ground-dwelling hypermetabolic insectivore stripped of its leaf litter cover stood zero chance of surviving an encounter with an ant supercolony. Conservationists responded to the ant threat with a targeted strategy of indirect biocontrol. In 2015, scientists introduced a Malaysian microwasp designed specifically to parasitize and destroy the scale insects. The logic was simple. By deploying the wasp to eliminate the scale insects, conservationists severed the honeydew supply. Starved of their primary carbohydrate source, the crazy ant supercolonies collapsed. But while the systemic intervention against the ants worked, tracking the primary victim of the cascade failed. In the late 1990s, a massive physical survey utilized 4,150 trap nights across 15 different sites. The traps yielded exactly 0 shrews. The methodology itself was incompatible with the compromised environment. Heavy environmental noise ruined the data collection. Massive robber crabs crushed the physical equipment, while residual crazy ants swarmed the bait stations, making it nearly impossible to capture a living shrew. The ecological interventions successfully stabilized the forest floor, but the inability to accurately monitor cryptic species meant scientists were operating entirely blind. The difficulty of tracking the species created decades of administrative hesitation. After being assumed dead since 1908, biologists clearing a track in 1985 accidentally captured 2 live shrews. The species had survived the cascade in microscopic pockets. Those 2 individuals died in captivity shortly after, but their brief rediscovery alerted scientists to the danger of committing the Romeo error. This is a conservation paradox where prematurely declaring a species extinct causes funding and habitat protection to cease, thereby ensuring the species actually dies off. To resolve this tension, researchers in 2023 applied the IUCN's Records and Surveys model. This formula calculates extinction probability by combining historical sighting frequency, the intensity of modern search efforts, and the base detectability of the species. To feed the model with definitive data, researchers analyze the ultimate proxy evidence, the stomach contents of hundreds of feral cats culled on the island over several decades. Feral cats are highly efficient hunters of small mammals. If a shrew population existed, a cat would have likely consumed one. After analyzing hundreds of stomachs, researchers found 0 trace of shrew DNA or bone fragments. Plugging this absence into the model returned a 96.3% probability of total extinction. This provided the objective mathematical justification necessary for the IUCN to officially close the book on Crocidura trichura. The extinction resulted from a compounding sequence of systemic failures: a pathogen spillover from maritime shipping, a trophic collapse driven by the loss of native predators, and the invasive synergism of the yellow crazy ant. The primary lesson from Christmas Island is that relying on the visual confirmation of cryptic species in a degrading environment is often insufficient for effective protection. Modern conservation requires the proactive deployment of robust biosecurity protocols and non-invasive monitoring. Analyzing environmental DNA in soil samples and utilizing autonomous acoustic recording units allows us to track fragile populations long before visual traps are compromised by environmental noise. Treating an isolated ecosystem as an interconnected mathematical system and mapping the mechanics of its failure provides the clarity needed to intervene before the next island species fades into silence.